Dierssen. I work in the Center for Genomic Regulation in Barcelona. And we basically are interested in the neurobiology of Down syndrome, but also in how non-pharmacological treatments can help these people to overcome the cognitive deficits.
Great. And so can you give some examples of what those might be? I understand that you have a clinical trial going
yes.
currently. Maybe
so the first thing that we did was to try to understand how was the brain changing upon environmental enrichment, so that what would be the parallel in mice for early intervention programs in humans, and how did this work in animal models. This gave us some cues about which could be the best candidates to go for to try to boost this neural plasticity that was going on in the animal models. So one of our best candidates is called Dierssen. It's a kinase that has been involved in different processes that are important for the construction of the brain. So this means for the creation of the dendritic arborization, for the organization and the topology of the connections. And this is very important to understand how information is processed in the brain. So this was our first work. And very luckily, one inhibitor, one normalization tool for, let's say, returning Dierssen activity to normal levels was something that is in the green tea, it's a polyphenol, and this was discovered by a French group, the group of Lejeune. So we decided to try to boost plasticity in our mouse models with this agent, and it worked fantastically. So it really improved very much the effects of environmental enrichment, and they were more stable. And so we decided to go for a clinical trial because it was a nutraceutical, it was safe. Because one of the problems with all the things that we are discovering is how to put this into humans. Yes. It has to be safe, it has to be something that can be administered chronically, and it has to be something that really does something, that's some effect that you can see. Mm-hmm. So we decided to go for a clinical trial, as you have said. Yeah.
So are the environmental factors that you're working with strictly dietary, or are there other sensory issues that you're working with?
We use a model in which what we do is we stimulate different sensory, cognitive and motor areas. So basically you put big numbers, social groups of animals, of mouse models, and then you administer them changes in the environment. Olfactory changes, visual changes, changes in the composition of the environment. But also you give them the opportunity to train, to do exercise, motor exercise. And also we put them like cognitive problems. It's like if the mice would go to school to learn.
So what would be the optimal age, do you believe, in a human population? Where is your clinical trial situated in terms of age?
I mean, one is where we can act, and the other thing is what would be the best. In my opinion, if you want to really make a difference, it's better to act in the moments in which the brain is being really grown up and the connections are being made. So the postnatal period for most of the regions of the brain is very important. The problem is that by now this has more difficulties in practical terms. So ethically it's more difficult to obtain approval for a postnatal treatment. So we first had to go to young adults. But this is a very good stage also, because you can see the problem from two sides, no? One can be, okay, let's build better this brain, but the other can be, okay, we have already a lot of population there. Can we help them also? So this was the idea of the second, of this trial, no? Of the second more human study, to try to see if we really can restore or rescue a phenotype that is already there. Because this phenotype comes from a bad construction, but also from a bad plasticity. And in fact, we have positive results, so we are very happy because it has been quite successful.